Hey everybody, in this video we will talk about Sanger sequencing, which is a really common technique in genetics. And essentially, if you want to determine the sequence of a DNA molecule that is like, you need 1,000 base pairs sequenced, you would use Sanger sequencing. We talked about some other technologies for genome sequencing, and yeah, you wouldn't use those technologies for a simple 1,000 base pair molecule. You would use this, and this is pretty cheap. You can get this done for like $5. So labs around the world rely on Sanger sequencing for most of their sequencing needs. Okay, so how does this process work? Well, it's a rather old process, a couple decades, and it has gone through some changes. So we're going to talk about the classical Sanger sequencing method and some relatively recent advances and that would allow you to see how it is done today. So you kind of need to understand how PCR works. Some of the concepts are the same, so make sure you understand how PCR works by watching the previous video. The PCR video was V2201, I believe, and this one is going to be V2202. So, okay, so Sanger sequencing. Well, let's have a piece of DNA here, and let's say for Sanger sequencing to work, you need to know part of the sequence. You need to know the sequence for part of it. So let's say this is the known region of the molecule, and then the rest of it, okay, let's say this is unknown right here, and let's say this is about 1,000 base pairs, and this region over here, we'll say this is 20 base pairs. So we can use Sanger sequencing to determine the sequence of this part of the molecule. So a Sanger sequencing reaction needs a couple different components. It needs a primer, a single primer. So PCR requires 2 primers. Sanger sequencing requires just 1 primer. And that primer obviously is going to have to bind to the known sequence. We also need Dntps, DNA polymerase, template DNA. So the template DNA is the thing that we need to sequence. Buffer for the polymerase, and water, and we're almost done. We need one more thing. So all these components are in a PCR reaction, right? And so this is identical essentially, except we only had 1 primer. Now the template DNA is going to be present in thousands of copies, so thousands of copies here. And the polymerase will probably have thousands of individual proteins here and tens of thousands of Dntps, and also thousands of copies of the primer, all in a PCR tube or sequencing reaction tube, which is about 200 holds about 200 microliters of volume. Now, number 7, the 7th component, we are going to need Dntps. Specifically, in the classical version, we need 1 Dntp. I'm going to talk about this in a second. In the new version, well, we can use all 4. So NTP stands for A, C, G, or T. So with the classical version, we're going to need to perform 4 separate reactions. 1 reaction with DDATP, 1 reaction with DDCTP, and so on and so forth. You'll see why in a moment. So if you did watch some of the videos in Lecture 21, you know what Dntps are. But let me just go over those briefly, just in case you don't remember. So Sanger sequencing is a form of chain termination based sequencing. So chain termination based sequencing. And Dntps are chain terminators. So let's look at a Dntp. So these are standard deoxyribonucleic acid triphosphates. So that TP, that's the triphosphate. So I'm going to say 3P there for triphosphate. That's attached to the 5' carbon, like we've been talking about all semester. Here is the deoxyribose sugar molecule. So 5 carbons, 1, 2, 3, 4, 5. This is the 1', 2', 3', 4', 5'. Now, deoxyribose or deoxyribonucleotide, a deoxyribonucleotide has deoxyribose, and that's this 5 carbon sugar. And the deoxy, the 2' deoxy is this thing right here. There's no hydroxyl group here. There's just a hydrogen on that carbon. Here, there's a hydroxyl group on this 3' carbon. So this is the deoxyribose sugar. I didn't put the other hydrogens on here like I never do, right? You know that by now. And then here's the base, which can be A, C, G, or T. Now this is just the standard structure of a deoxyribonucleic acid triphosphate. And this is what DNA polymerase uses to synthesize DNA. So Dntp is almost identical. So let me quickly diagram this. I'll just put, instead of putting the carbons, I'll just put the little angles, meaning that's a carbon. There's the base up there. So there's the H for the deoxy part. Now the okay, I should say this, if you don't remember from the previous video. DD is dideoxy ribonucleotide triphosphate. So the Dntp was for deoxyribonucleotide triphosphate. The DD is for dideoxyribonucleotide triphosphate. So the only difference is this thing right here. This is the 3' carbon. There's no hydroxyl group attached here. You can put a hydrogen. You can put anything except the hydroxyl group. And you'll see towards the end of the video, sometimes you can make these fluorescent. You can attach fluorescent molecules to a DDNTP, and some biotechnology companies add that fluorescent molecule. They attach it here to this 3' carbon. But the DDNTP is called a DDNTP because there's no hydroxyl group here. So we'll keep it simple. We'll put 2 hydrogens, one here, one here. So no hydroxyl groups. Now these are known as chain terminators because, as we have talked about throughout the semester, DNA polymerase synthesizes DNA by adding a nucleotide to that 3' hydroxyl, right? So that 3' hydroxyl is used to add the next nucleotide in a growing chain of DNA. So you get rid of that OH, and then no more nucleotides can be added to that chain of DNA. So it stops elongation or stops DNA synthesis, terminates chain elongation. So these types of NTPs, DDNTPs, are going to be important for the Sanger sequencing reaction. Okay. So now how does sequencing work? Sanger sequencing work. Just briefly, okay, so remember that initial diagram I started with say a 20 nucleotide or base pair known region of DNA and 1,000 base pair unknown region that we want to determine the sequence of. For this video, let's shrink this up. Let's assume this 20 nucleotide region is just 3 nucleotides long. I'm going to put CAT there and GTA up here. And this is the 5' end and this is the 3' end. And for the unknown, we're going to pretend we know it, right? We're going to pretend we know it so I can teach you how Sanger sequencing works. So okay, so there's the unknown sequence, unknown in quotation marks, right? And this is the top strand. Okay, now it's pretty short, but let's imagine it's 1,000 base pairs long. And we don't know the sequence, but for me teaching you how Sanger sequencing works, well, we'll secretly know that this is the sequence. So okay, so now we'll see how Sanger sequencing works. Okay, we're going to run a reaction, sequencing reaction, to determine what this sequence is, right? Because we don't know it. We don't know what that sequence is. Okay, so what happens? Well, all those components that we talked about previously, primer, Dntps, DNA polymerase, template DNA, buffer, water, all that's in the tube with one Dntp. So that's all in the tube. What we need to do first is denature the DNA. So imagine there are thousands of these molecules. This is the template DNA, and we're denaturing the DNA in the tube by heating that tube to like 95 degrees Celsius. So you leave it there for like a minute. And then you lower the temperature. We could do this in like a PCR machine or a thermocycler that we talked about in the last video. And so denature means what? We split the strands apart, right? By breaking those hydrogen bonds. So now the reaction is full of single-stranded DNA. So let's say we'll put denature here. And then what do we need to do? We need to anneal the primers. So the primers, well, they're usually about 20 nucleotides long, but here, in this example, they are only 3 nucleotides long. And this would never work in reality. It has to be much longer. 3 nucleotide primer, it wouldn't work for various reasons. But we're assuming it's for our example, 3 nucleotides long. In reality, it would need to be about 20 nucleotides long. So we're going to lower the temperature so these will stick to the primer binding site. So thousands of primers, thousands of templates. Let's put one of them here. So I'll show one interaction between the template and the primer. Okay, so it's the 5' end here, and this primer is going to have an OH group, so DNA polymerase can add a nucleotide to the end of this. You can synthesize the other strand. Let's say the annealing is done at, let's say 55 degrees, 55 to 60 degrees Celsius. Well, you know what, we'll keep it consistent with PCR. Let's say 60 degrees Celsius, and that would change depending on the annealing temperature or the melting temperature of the primer, like we talked about in the last video. Okay, so now we need to extend. And how do we do that? Well, we can raise the temperature to 72 degrees Celsius, and that's the temperature that the polymerase likes, and the polymerase can start elongating this chain. So I want to put a couple molecules in here. So remember, the test tube has many copies of the template, say thousands of copies of the template. And we made them all single-stranded by denaturing. And when we annealed all of them, we assume all of them have the primer annealed to the primer binding site. So let me diagram just 3 copies of the template. And now notice how I'm not diagramming the top strand of the template, right? Because we don't care about the top strand with sequencing, because we only added 1 primer. And this primer, it only binds to the bottom strand. So the top strand's floating around in the reaction and it's not really doing anything. Okay, so here I have just 3 copies of the template annealed to 3 primers, and this represents the thousands of copies of the template and the thousands of primers that are nailed to those template copies in the reaction. Now, inside our reaction, just briefly, what do we have again? Water, buffer, DNA polymerase, 1 primer, the GTA primer. We don't have a second primer in there. Dntps, so all of the deoxyribonucleic acid triphosphates, all of them are in there. So that's ATP, DATP, DCTP, DGTP, DTTP, and one DDNTP. Let's go with DDATP to start. So one thing to keep in mind is that for the DNTPs and the DDNTP, whichever one it is, these are found at much higher levels than the DDNTPs. So let's say for each of these, DATP, DTP, DCTP, and DGTP. Let's say each one is present at 10 micromolars. You don't need to memorize that number. But that's a common concentration for a nucleotide triphosphate in a PCR reaction or a sequencing reaction. Now we could say for this reaction, we're going to choose DDATP, and we're going to use it, let's say, at 2.5 micromolars. So none of the other DDNTPs are present. So just DDATP, and it's present at about 1/4 the level of the DATP. Okay, so all that stuff is in the tube here, and we're at the extension step. So the polymerase is going to bind to the DNA in the primer, and it's going to try and elongate the primer or extend the primer. So the first thing it's going to add is a C. Then it's going to add a T, and then it's going to add an A. So what are the odds it's going to add a regular DATP or a DDATP, right? There's a slightly higher chance it's going to add a regular DATP. Okay, so we'll put an A in there. And then we'll go again, okay, C, C, oh, there's another A here. Okay, let's say this time it added the chain terminator. Okay, so I'm gonna put my chain terminators in this red color here. Okay, now it put the chain terminator DDATP here. So polymerase can no longer extend that chain. Okay, so so this one, the polymerase is the polymerase down here, extending this one, the same time there was a polymerase working up there. Okay, let's see what happens here. And let's say this time, the very first time, the polymerase inserts an A, inserts the DDATP here. Okay, so this chain is terminated very early, right away, when it comes across with the first A is needed. And let's look down here. Okay, polymerase down here, synthesizing. Let's say, oh, okay, we incorporated 2 normal A's here. Well, okay, say it's this one right here. This A here gets the chain terminator. Okay. So now what we can do to understand how this process works is count up the number of nucleotides in each single strand here. Each newly synthesized strand. Don't worry about the template, just count the lengths here. We can see there is, this one's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 nucleotides long. This one is 1, 2, 3, 4, 5, 6 nucleotides long. This one is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 nucleotides long. Okay, so in this reaction tube, if we were to separate these strands, we would have single-stranded DNA molecules of 9 nucleotides 6 nucleotides, and 15 nucleotides in length. Okay, so let's take a look at what another reaction tube would look like. Remember, we need to do 4 reactions to do the sequencing, to sequence our target DNA. Each reaction has to contain a different Dntp. So we just went over DDATP. Let's take a quick look at another one. I think I want to take a look at CTP. Okay, so let me briefly put in 4 copies of the template, or quickly put in 4 copies of the template. Now keep in mind there are thousands of copies of the template in this reaction tube. And I hope this helps you understand Sanger sequencing. This is essentially how I describe it in the notes. So if you read the notes, you probably know what I'm doing here. If you didn't read the notes, then reading through the notes later is going to be really easy for you. So I guess I'll put the 5' and 3' in. So same setup as before. The primer is annealed to the templates. And this time we are working with DDCTP, which is found at a lower concentration than is the analogous DCTP. And keep in mind, all the other DNTPs are there. It's just the DDNTP, which is only present in one form, and that's DDCTP in this reaction. So when we're sequencing DNA by the classical Sanger sequencing method, we need 4 reactions. One has DDATP added, one has CTP, one has DDTTP, and one has DDGTP. Just to, if you wanted to mention that, just in case you're a little confused. So, okay, so we're doing CTP now. Now let's take a look here. Let's say, oh, right away. Let's say a DDCTP is integrated right away, or inserted right away into the growing chain. Okay, so this chain was terminated really early. So that's 4 nucleotides long. Okay, let's go down here. And let's say a regular C is put here and here, and then a chain terminating C is put in. So this one, 1, 2, 3, 4, 5, 6, 7, 8, is going to be terminated after it is 8 nucleotides long. I'm including the primer in the length, if you didn't realize that already. So 1, 2, 3, okay, let's try another one here. We haven't done this one yet. Okay, there's a C there. So the C's are being put in randomly, and the probability of a C being put in is, dependent on the concentration of the DDCTP relative to the DCTP. But for this, let's just assume that the regular CTP is found at higher concentrations than the DDCTP. 1, 2, 3, 4, 5, 6, 7. I'll use the red. And let's do one more. Okay, let's assume all regular nucleotides, or C's, all the way up to here. Okay, let's count this up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So this one's 13 nucleotides long. Okay, now we'd have to do 2 others, but I think you see what's going on here. The same thing would happen with the others, but the lengths of the DNA would be different depending on the positions of, if we're doing G next, then the positions would change depending on the location of the C's, right? I think there's only one C in here, so there'd be only one DNA molecule synthesized if we put in DDGTP. I think, well, you know what? I'm going to address that later. Okay. So, and we would need to do TTP also. So, okay, we have these 4 reactions. Now what we need to do is perform gel electrophoresis. And we haven't talked about this yet in this course. I know you talk about it or maybe even run some gels in BSc 197. But let's review. So gel electrophoresis is used to separate DNA molecules based on their length, right? So we have two types of gel electrophoresis that are standard. One is called denaturing. We use this word a lot, right? Denaturation. So you can probably guess what's going on in that type of gel electrophoresis. And non-denaturing. So the denaturing gel electrophoresis is for analyzing single-stranded DNA. Non-denaturing is for regular DNA. Denaturing gels or gel electrophoresis also works well for RNA. So, okay, how does this process work? So, just in case you don't remember anything about how gel electrophoresis works from previous courses. So, let's say this is a semi-solid gel. Semi-solid, so it's like gelatin. Like gelatin or jello, but it's usually made of a gelatin-like compound called agarose. And these are little sample wells where you load your DNA solution into these little wells right here. And you apply a current, and this is submerged in a buffer, and one can apply a current. Let's say we load the DNA here, and let's say that the negative charge is over here, there's a positive charge over here, and the phosphodiester backbones make DNA have a slightly negative charge, so they tend to migrate towards the positive charge over here. And so if you load DNA, let's say many molecules of DNA in here, and they're all different lengths. Well, the shorter ones move through faster than the longer ones. So the shorter ones race through at a faster rate than the longer ones. So you end up separating the DNA molecules by length. So the longer ones end up closer to this side of the gel, and the shorter ones end up down here. And why do they go through at different rates? Well, if you were to look at this little gelatin semi-solid-like material with a microscope, what you would see is all these little pores. And the longer ones have a hard time getting through these pores and migrating through the semi-solid material. Harder time than the shorter ones do, right? You can imagine the longer ones will get tangled up and have a harder time getting through. Okay, so that's essentially how gel electrophoresis works. And I guess long story short, it's going to allow us to separate the DNA based on size. And that's going to be important for Sanger sequencing. So, okay, so let's say we have our sequencing reactions here. Now, we went over the size of the molecules, synthesized DNA that we would see, that we should expect in the reaction with the dideoxy ATP and the reaction with the dideoxy CTP. We calculated a 9, a 6, and a 15 nucleotide molecule, 9 nucleotide, 6 nucleotide, and 15 nucleotide for the dideoxy ATP reaction. And for the C, there were 4 molecules. One was 7 nucleotides, one was 4 nucleotides, one was 13 nucleotides, and one was 8 nucleotides. Okay, so now when we do our reactions here, we don't know the lengths of the DNAs inside that reaction. What we need to do is do the gel electrophoresis here and determine, what type of DNA molecules or what the lengths are of the DNA molecules in that sample. So I'm going to put 5 lanes in here. And each lane is going to get a different sequencing reaction. So I'm going to put DDATP here. I'm going to put the dideoxy-CTP reaction here. And, you know, we also ran the GTP and the TTP. I'm going to put T right here reactions, but we didn't go over them on a piece of paper. Now over here, I'm going to put what's called a DNA ladder. Now, a DNA ladder is, you know, you can buy a DNA ladder from a biotechnology company, and essentially what it is a solution of DNA molecules where each molecule has a known length. Like the biotechnology company synthesizes the DNA or makes it so there is a molecule of a certain size over a size range in the solution. So maybe I should just explain that a little better. Sorry about that. So if let's say we ordered a DNA ladder from a biotechnology company, and this ladder has from 14 nucleotides to 6, has molecules of 4 nucleotides long, all the 5 nucleotides long, 6 nucleotides long, all the way up to 16 nucleotides. So all those molecules are in this DNA ladder. So when we run this DNA ladder here, we can see a DNA molecule of every size from 4 to 16 nucleotides on there. And that's going to serve as a good reference for the molecules in these reactions right here. So, okay, we're going to load all our DNA samples into these wells, and then now this is floating in a buffer, and the DNA molecules are going to migrate through the gel here. Now, we don't need to worry about how so once they migrate through, we can take a look and see where the DNA is in here. We don't need to worry exactly about how that process works. We have dyes we can stain the DNA with. You could use radioactive nucleotides in this process, and so you can use detect the position of the DNA based on where the radioactive molecules are. So there are a number of different ways you can do that. So we're going to skip over all that, and we're just going to assume that, okay, now we ran the gel, we know where the DNA is. Okay, we can see where the DNA molecules are. So let me put the ladder in first. So I'm just going to put these little markers here to help me diagram somewhat neatly. So this little band right here, this is DNA. And why? You know, it's shaped like a rectangle here. When we look at it, why is it shaped like a rectangle? Well, because when we added the DNA here, you know, the DNA molecules filled this whole well. So when they migrated through, they maintained that shape of the well up here. So they form these like rectangular bands. And so, like I said, with this ladder right here, we ordered this special ladder right here, and this ladder has a molecule of length 4, 5, 6, all the way up to 16 nucleotides in length. So this is a great ladder. I don't think any companies actually sell a ladder like this, but we need one for learning how Sanger sequencing works. So we put this in here. So, okay, great. Now, when we ran our DDATP reaction, what do we see there? Well, we predicted we should see 6, 9, and 15. So let's go, this is going to be 4, 5, 6, 7. I'll put numbers in here so we can keep track. Okay, so we should see a band at 6. We should see a band at 9. We should see a band at 15. What about this one? Okay, so that is for the DDATP reactions. We see 3 DNA molecules, 1 at 6, 1 at 9, 1 at 15. And for this DDCTP reaction, well, we said we should see 4 bands, 1 at 4 nucleotides, 1 at 7, 1 at 13, and 1 at 8. Okay, so what about the others? Well, you know, I'll just jot down what they should have been if we had gone through it with a paper and pencil. So there should have been 5 bands for the dideoxy-TTP reaction, and there only should have been 1 for the GTP reaction. Okay, and that one should have been 14 nucleotides long. So for the dideoxy-TTP, we should see 5, 10, 1 at 11, 1 at 12, and 1 at 16. And for the GTP reaction, we should see 1 at 14. Okay, so great. So if we performed a Sanger sequencing, I guess, assay to determine the sequence of our molecule, we performed a reaction with dideoxy ATP, CTP, TTP, and GTP, so 4 reactions. We analyzed those reactions by gel electrophoresis, and we see all these different DNA molecules of different lengths. Now, to determine the sequence, what we need to do is look at the sizes of these bands right here. So we know the sequence of our primer, right? Our primer was GTA. And so we can determine what sequence comes next. So now we're in the unknown. Remember, unknown with quotes, because we don't know what this was and we're trying to determine the sequence of it. So we can determine what that next base is by looking at the shortest fragment here. The shortest fragment is in the DDCTP well, right? So it's 4 nucleotides long. And because it's here, we know that, okay, there must be a C right here. Now count how long this is. 1, 2, 3, 4. Okay, so that makes sense that this is 4 nucleotides long. This sequence, a primer, plus the single nucleotide that was added to the primer before and after which the chain was terminated, because it is a dideoxy CTP, is only 4 nucleotides long. So then we go to the next one. T. Okay, there's a T there. That makes sense. Okay, this is 5 nucleotides long. It makes sense that it ends with a T because it's in the dideoxy TTP lane. So okay, everything's great. So you see what we're doing here. We're following the bands up. So the band of 6 nucleotides long is in the ATP lane. Then C again, 2 C's there. And then A. Then we have 3 T's. Then we have a C. Then we go over to the G, then A, and over to T. Okay, so we've done our sequencing reaction. So this is the sequence of the non-template strand. And now we can determine the sequence of the template strand. So just by complementary base pairing rules, right? So we knew this sequence over here. We knew the primer bound to a CAT. And we didn't know this, but now we can determine it from this sequence right here, just by complementary base pairing rules. So there's a C here. I'm going to put a G, A, T, G, G, T, A, G, C, T, A. Okay. So we didn't know this going in, right? That was the unknown. And we did our sequencing reaction. We got our results. We interpreted the gel, and now we know the sequence. Let's see, does it match? Does it match the original? Where is the original? Let me see. What did you do with the original, Tom? Okay, here it is, the original. I hope this matches. It'd be really embarrassing. If it doesn't match, I have to start over. Okay, we can see, yes, this was the original. GTA, CCA, TTC, GAT. Yep, okay, great. So it works. Okay, so now you have an idea of how the classical Sanger sequencing method works. Okay, so on an exam, quiz, or anything like that, if you had a question like this, and this is actually a problem in the notes. Hope we're recording. Yes, we are. Here's our Sanger sequencing result, a gel from a Sanger sequencing assay. and I asked you to determine the sequence of the non-template strand, what would you do? Okay, well, I'll show you what you would do. You take the gel right here. Okay, we have this little diagram set up to make it easy. Now primers, I didn't show you the sequence of the primer, it doesn't really matter, but primers can only be extended from the 3' end in the 3' direction. So we know that's the 5' end, we know this is the 3' end. So look at the shortest band. We should put a T right here. Well, that is 4 nucleotides long, so that makes sense. We have 1, 2, 3, 4. Okay, 4. Okay, how about this one? This DNA molecule ends with an A, because it's the dideoxy ATP lane, and it's 5 nucleotides long. So that makes sense. 1, 2, 3, 4, 5. Okay, so we're just reading up. CCC, G, TT. So this is really easy, right? You probably don't even need to understand genetics. There's probably only really one way to put this data in that makes sense with this diagram. So maybe I won't give you a diagram like this on the exam or the quiz. Okay, so now we have the sequence of the non-template strand. Non-template. Now, to make the non-template, we need to copy the template, right? So determine the template just by complementary base pairing rules. Okay, so when I ask these questions on an exam, you're going to need to pay attention to the 5' and 3' ends, right? There's not too many questions I can ask that are like medium difficulty. They're either really difficult or they're like super easy. So you got to pay attention to the 5' and 3' ends, right? So 3' A, 5' C, this A end here, this is the 3' end. So if this was a choice, 5' C, C, G, A, T, T, A, C, G, 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 T, A, and the question was, what is the sequence of the template strand? A lot of students would say, oh, where is this? I don't see this. This would be one of the choices, a multiple choice exam, and they'd be like, oh, I don't see this anywhere. And they don't realize that they need to flip this around because, well, the 5' is over here, and over here on this diagram, the 5' is at the right side. So pay attention to these 5' and 3' ends, and note that you may need to reverse it to find the answer among a group of choices on a multiple choice exam. Okay, so that's how you would answer that type of question. Now, a couple more things on Sanger sequencing. So some advances. So that was the classical Sanger sequencing method. And you can take a look in the notes where we talk about how they use radioactive dideoxy nucleotides in order to be able to see the DNA on the gels after gel electrophoresis. But with some advances now, it's possible to add fluorescent molecules to the dideoxy NTPs. So let's say, and the colors, they might be semi-standard. I think each biotechnology company that produces components for these types of experiments might use their own color system. So we don't need to memorize the colors, so which color goes with which nucleotide. But let's assume for say dideoxy ATP, there's a red fluorescent molecule attached to that. TTP blue, CTP green, GTP yellow. Now, if we ran a sequencing reaction, or 4 sequencing reactions, why am I doing that? I don't know. So, and analyzed the 4 reactions by gel electrophoresis, well, then we should see different colored bands in here. We put a UV light on here or something, and we see the fluorescence. And we see red bands for ATP, blue for TTP, green for CTP, yellow for GTP. So let's imagine that we use fluorescent dideoxynucleotides for this example right here. Well, what would that look like? I'm going to cheat a little bit. I already diagrammed this out. It would look something like this. And how would you read the sequence? Well, you read it the same way, right? You read it the same way. Okay, so it starts with a primer. Now, let's say we don't even know how long the primer is, but we start with a T, A, then we're still going up by size, right? Going from shortest molecule to longest molecule. 3 C's, a G, 2 T's, 2 A's, a T, a C, 2 G's. Okay, and that would be the non-template strand, and then you determine the template. Okay, everything's great. It's easy, you know, it's pretty too, right? Better than these just, you know, dark black rectangles. And we get the same sequence. That's a no-brainer, right? Okay, that makes sense. So one of the advantages of using these fluorescent molecules here is you don't need to do 4 separate reactions. You can just do one reaction and add all of the dideoxynucleotides to the same tube. And then when you analyze your DNA on a gel, it looks something like this. Sheet it again here. Okay, so we have our code here. It's the same as the code up here. A is red, T is blue, C is green, G is yellow. So we can determine the sequence. So here's the primer. T is blue, so blue is I can put it like this. T, A, C, C, C. Yellow is G, 2 C's, 2 A's, C, oh, blue was what? Blue was T. C, so you can still make mistakes. G is yellow. Okay, so T, A, C, 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 G, C, C, A, A. Blue is T, right? Well, we have computers doing most of the interpreting of this, so less likely to make mistakes. C, I'm messing up here. T, A, C, 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 G, T, T, A, T, C, G, G. And it matches. Okay, so that's the non-template strand. We could do the template strand by complementary base pairing rules. Okay, great. Okay, so one more advance. So we don't have to run these gels anymore. There's an automated system. And what it is, you know, we still need humans to set it up, right? To put the samples in, or at least put it in a place where a robot can get to the sample. But it's called capillary electrophoresis. And it's a lot like gel electrophoresis, but the gel material is inside a little capillary tube. So here's a capillary tube. And the process works the same way. So this is full of the agarose gel-like material, and the shorter molecules are going to go through faster than the longer molecules. So essentially it's separating the DNA molecules by size. So you have DNA molecules, a mixture of sizes inside a sample. You load the sample in and it separates the molecules by size, with the shorter ones coming through first. And this is a little window right here. And there'll be a little laser. And that laser will shine on the window, and there'll be a detector here to detect the fluorescence. And so just like in the previous example, so red, we'll use red for DDATP, blue for DDTTP, green, and yellow for a DDGTP. So when the DNA molecules come through, now they've all been terminated by a different dideoxyribonucleotide, and they'll glow a certain color depending on what dideoxynucleotide they were terminated with. So, and the detector is hooked up to a computer, and it produces what is called a chromatogram. And sometimes the DNA chromatogram. On the x-axis, essentially, is time, right? And up here is, say, early. Over here is late. So the first molecules coming through are the shorter DNA molecules. And on the y-axis, we call this fluorescent intensity. Fluorescent intensity here. So, and what the computer is going to detect is the 4 different colors. So the 4 different fluorescences. So we're going to have 4 different colors being registered on this chromatogram here. So using our previous example, would be the easiest way to set this up here. Keep everything. So in our previous example, right? So if we did gel electrophoresis, the gel would look something like this. If we were using capillary electrophoresis, well, okay, so this 4 nucleotide molecule would be coming through first because it's shorter than the others. And then we see blue. So the first thing that comes through is blue. Okay, so we have this blue like peak right here, okay, because the blue intensity was very high. And then the next time we see blue, it's not going to be for a while. I'm going to put little marks in here so I can make a nice neat chromatogram for us. So this is 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, so the next time we're going to see blue is going to be way down here at 10 and 11. Oh, so we get a blip at 10 and a blip in blue at 11. Okay, so it's not like it's going to do all the blue first and then do other colors next. It's still, the whole time, it's going to be looking for red, green, and yellow colors too. And over here, we see that there should have been a 5 nucleotide long sequence that ends with A, which is red. So we should expect to see a red blip at about this position right here. So the red intensity was really high at about this time point right here. And then we're not going to see another one until 13. So the red signal is going to be really low until 13 or 12 and 13. So it's going to look something like that. I'm a little off scale here, but I think you get the point. Now, okay, it's also looking for green the whole time. And then at, the second, third, and fourth molecules through are all going to be fluorescent green because they were terminated with a green DDCTP. So it's going to look like this. And then there'll be another one way at 15, way down here. And the last color it's looking for is yellow. And so it should have seen a yellow at about 9, 16, and 17. So we should see a blip of yellow right about here. Then not another one until we're off the screen here. Okay, so this is what a DNA chromatogram looks like. And essentially, when, like in the lab here at ISU, we send our sequencing off and they actually sequence them at a sequencing facility at the U of I. It costs like $5. And what they send us back is, this right here. And a computer usually reads it for us and just gives us our sequence. But we can take a look at the chromatogram and then we can see that, okay, we know what the colors mean. So we can see that, okay, so the first one, T, then the next one, A, and then we have, 2, 3 greens there, so CCC, then a G, then 2 blues, 2 reds, and then a green. And so, okay, that would be our sequence of the non-template strand. And then that's the sequence of the template strand. We're going to put our 5' and 3' ends in here. Okay, so that's how you read a chromatogram, and that's how those chromatograms are produced. So I hope you understand essentially how Sanger sequencing works. The classic Sanger sequencing method that, you know, did 4 separate reactions for each dideoxyribonucleotide. Required, I think, radioactivity usually to detect the positions of DNA on a big sequencing gel. I think when I did my master's degree back in 1999, I think it was 1999, I was probably, that was right when people were switching over to doing this capillary electrophoresis, and I actually got to do some big sequencing gels, and it was not a lot of fun. It was really expensive, and it was a lot of work. So this is much easier. Okay, so that's it. That's how Sanger sequencing works. There are some problems in the notes. If you have any questions, make sure to email me. And other than that, I will see you in the next set of lecture videos. That will be lecture 23.